Okay, Chapter 12, Cell Reproduction. So there we are. So, we've talked a bit about mitosis and meiosis in the past, and we need to talk about those again and add some more detail into all that, and this idea of the replication fork, and sources of mutation and change here. One of the big themes we're going to be talking about all semester is variation, and this is where a lot of variation stems from. So we'll talk about variation as well. Cells essentially replicate, copy themselves, for three things. They need to restore the ratio between the size of the nucleus and the amount of cytoplasm present, so non-nucleus. This sort of limits how big a cell can be. That ratio is really important. And a cell can only grow to a certain size and then it has to replicate. That's all about diffusion rates and osmotic gradients and things. There's also the need for the growth and development of the individual. And of course, you guys are just coming to the end of puberty, as we often talk about, and your growth and development is slowing down now. And you're entering that phase that hopefully I'm still in, of maintenance and repair. But of course, past 50, maintenance repair starts to slow down too. So I may well be in a situation where I'm not maintaining and repairing my cells as efficiently as I once did. Then hopefully you guys definitely are. I'm still not, aren't I? So we talk about, when I was a year 12 student, Interphase was basically the bit before, but straight after cytokinesis, all the way around to Prophase and Metaphase, and Mitosis is a bit we were really interested in. And that knowledge has changed, or at least knowledge for the year 12 students has certainly changed. So then now we talk about three stages within Interphase. Interphase used to be considered the resting stage, and it's certainly nothing of the sort. So the first stage is pre-DNA synthesis, often called growth 1 or gap 1, so G1. And in this we produce a whole lot of proteins and enzymes and collect all the materials we'll need that we're going to require for synthesis, stage 2. In stage 2, every single chromosome is copied. So you've gone from having a cell with 2 sets of chromosomes, a diploid cell of 2n, to a cell now which has 4 sets of chromosomes. So it is now tetraploid. It's 4n. And then the third stage is post-DNA synthesis, so it's G2, it's the second growth stage. And here we now start to gather together all the materials that are going to be required for mitosis. And for 2 cells, because the outcome of this will be 2 new cells that are identical. Well, essentially identical. So we've got gap 1, S, gap 2, or G1, S, G2, and these 3 stages are by no means resting, but that's interphase. During interphase, of course, the important thing is the cell continues to function. So it continues to read its DNA and produce proteins to maintain the cell and to participate in the whole organism. So it's a pretty busy time. Mitosis then occurs in 4 definite stages. And we talk about prophase, metaphase, anaphase, telophase. And you're going to need to remember that. And you're going to need to remember them with an image in mind. We'll go through each in a bit more detail. You can see here, there's quite different stages to what's happening with the DNA, which allows us to determine what phase you're in. And you're going to need to be able to recognize that. It's not unusual for you to be asked to recognize that. So in prophase, the chromosomes condense. So they should become short enough and thick enough, more to the point, to be quite visible under the light microscope. And you can see here the 2 copies. There's actually 4 strands of DNA there. Each chromosome is, of course, double-stranded, and the 2 strands are called chromatids that are held by a centromere. This is during mitosis. We name them differently. Then, these chromosomes, these pairs, line up across the equator of the cell. And the centromeres are used to, in fact, attach to spindle fibers. And we can't see the spindle fibers in this microscopic view, or vaguely. There's vaguely centrioles at each end of the poles, and they're connected through the equator by these spindle fibers, and these guys attach to the spindle fibers. And so metaphase is marked quite clearly by that stage, seeing all those chromosomes lined up across the middle. we then move to anaphase, and the two halves of the chromosome are going to separate and go one to each pole. So the centromere actually splits in half, allowing it to occur, and they follow the spindle fibers. Spindle fibers retract into the centrioles and take the chromosome with them. And again, so in anaphase, you get that really clear, distinctive feeling of them pulling apart. And then in telophase, they start to condense again. The spindle fibers disappear, centrioles disappear, and two new nuclei are starting to develop. And if you look carefully there, you can see a cleave. There's a new cell membrane starting to grow. And it's much more clear here. We've got two definite nucleuses, definite cytoplasm for each of them, and you quite clearly can see that cleaving where the new membrane's growing and we're going to split apart. And they are said to be essentially identical. And so we're back to having 2N. 2N here and 2N there. So one cell at 2N, at diploid, created a cell of tetraploid, 4N, and then we split that up to create two lots of 2N again. Because a cell needs to be two sets of chromosomes most of the time. And it's called cytokinesis at this point here. So meiosis is a second stage. And meiosis only occurs in gonads, and it creates gametes. So gonads, testes and ovaries, gametes, sperm and egg. In plants, we're looking at pollen and ovules. But again, occurring in the gonads of the plant, essentially, which is the flower bud. And the big difference here is, again, we've got this first stage, which is identical to mitosis. We go through G1, S, G2, and create an individual which is now 4N. That goes through mitosis, cytokinesis, and we end up with two cells that are now 2N, and then we do a second stage. So instead of condensing completely, these then realign themselves on the equator at metaphase 2, and are pulled apart to different poles and divided. So these four cells are 1N. They're haploid cells. They have one set of chromosomes. And these are sex cells, these are gametes. And there's a little animation there that I pinched from somewhere. I've forgotten where I even found it now. And this little spermia rise, and in 7 months we've got a new person growing. During the first stage of meiosis, homologous pairs arrange on the equator, and this allows for these things to cross over. So if you think about it, we have 42 chromosomes. No, we don't. We have 46 chromosomes. We have 23 pairs. Having made that into 4N, I now have 92, that's where the 2 came from, 92 chromosomes lined up across my cell. This minuscule little thing now has 4 meters of DNA chunked up into chromosomes arrayed across its equator. They get to intertwine. As they come apart, I can't do it with my hands, bits break off and reattach themselves. And so we can recombine the DNA. So this allows for crossing over, recombination of two adjacent pieces of DNA. And so the cell still gets the DNA, but it puts it somewhere different. And that can lead to quite different gametes in the next person, the next child, the child you have made, because of rearranging where we find the genes. So crossing over is quite important, because it does actually create this wonderful variation, particularly in gametes. Crossing over within ourselves occurs all the time, but it's irrelevant. So in my hand, there's probably lots of cells that have crossed over DNA. Particularly on my skin. I should say the skin that's facing the sun has lots of mutation because of the effect of the sun. So there's probably lots of crossing over and other mutations showing on my hands. But I can't pass it on. I'm stuck with that. That's my problem. If it happens in mitotic division, so it happens in the production of gametes, these changes get passed on. And we look around our classroom and recognise 13 different, or 14, including me, different people. We are all mutants. We're all different. And one of those areas of variations is crossing over right here. This thing here is called a chiasma. And you might see the term chiasma from time to time. The other thing that can happen, of course, is that we can cause one... We've got 2 chromosomes left there. And if they don't separate and go to different poles, but rather they go to the one pole, then, for instance, our red guy here could have it's going to get anyway, sit still. They can end up with 2 copies of the chromosome, and the other gamete, no copies of the chromosome. When those gametes mix with gametes from another individual, that can lead to a situation where you have 3 copies of the chromosome, we call it trisomy, or 1 copy of the chromosome, monosomy. And these, of course, create more variation. Some of it deleterious, some of it potentially beneficial, some of it is quite benign, you wouldn't even know. But essentially, it's not a good thing to have 3 copies of the chromosome. You have too many copies of that gene then. And the classic polysomy is trisomy 21. And we all be aware of Down syndrome. So here's this high-functioning Down syndrome girl, I would suggest, just by looking at her, who has those distinctive Down syndrome eyes. You see the poor body formation. So they have poor muscle tone, poor skeletal development, so shorter of stature, definitely delayed intellectual development, but it's only delayed. It can, in fact, in high-functioning Down syndrome people, develop. This is amount of time, which tells us what happens on chromosome 21, of course. There's a whole lot of genes we now know where to look for. But monosomy 21 doesn't exist. If you're monosomy 21, you don't get born. You miscarry. So clearly, you can cope with having 3 copies of 21. You can't cope with 1. That's interesting, isn't it? Polyploid is a name given to individuals with more than 2 sets of chromosomes, completely. Now, this doesn't tend to occur in highly evolved animals like ourselves, because that would probably abort. If that did happen, you'd have an abortion, a spontaneous abortion, miscarriage. But it does occur a lot in plants. So we often find plants with 3, 4, 5, 6, 8 sets of chromosomes, which is pretty messy. But a lot of their growth and development, of course, is asexual. They don't need to produce gametes, so it's not a big deal. And of course, many animals, very simple animals, worms, things, can use asexual reproduction also. So they can often carry 3 or 4 sets of chromosomes without any real problem. But you can't make gametes in that situation. So, genes and development, how many have time? Oh, running out of time again already. Homeotic genes. Homeotic genes are really important. They're the genes that actually control your development, laying down each level of tissue as we need to. We call these the derms. Endoderm, mesoderm, ectoderm. So the ectoderm clearly is the outside, the endoderm is our interior, and the mesoderm is in between them. And they get laid down and controlled by these homeotic genes. And they ensure this happens correctly. Cells specialize. So as we get older and older, our cells start to develop, as this is in utero, develop into these cell types. And I can't remember what number it is, 42 cell types? I can't remember. Some large number of cell types throughout our body. But they all start here. So it's that blastocyst. It's about 16 cells. And at this point in time, every single gene is switched on. And as we move through, we start to differentiate these genes into the mesoderm, endoderm, ectoderm, and then within those they have particular roles, epithelial, liver, whatever, and we end up with highly differentiated cells which have particular jobs to do. So these have lots and lots of different genes switched off. Here, they're all switched on, which is the great interest in adult stem cells, embryonic stem cells. Adult stem cells also exist within major organs. There are stem cell centers, but they have some stuff turned off. They're not as useful as embryonic stem cells, which I've just discussed, so I won't linger over there. So adult stem cells, already programmed to make particular cells, won't grow indefinitely, can be difficult to obtain or painful to obtain, and generally they make one cell type. Embryonic stem cells, unlimited, fantastic, but you got to kill babies to get them. Some people have a bit of problem with that. Cells will divide indefinitely. They can grow them in large quantities. You can then differentiate them to what you want to do with them, although we're not quite on top of how you do that. So embryonic stem cells essentially are better things to work with. The scientists are finding ways to work with adult stem cells to overcome that. And we've talked apoptosis often enough. But, just on the side, if mitosis goes wrong, apoptosis is the end result. So if something goes wrong in copying that cell, it just destroys it. Oh, and I just snuck it in within 15 minutes. How's that? I'm getting better at this.